Hi, today we are going to discuss about one of the most complex topic, nervous system. In this nervous system, coming to the point, I'm going to discuss about the terms which is tract and ganglia. Most of them are confusing with these two terms, even me too, obviously. And what is tract? Tract is nothing but axons of CNS. And this is not only axons, this is bundle of axons of CNS. This is tract. And what is ganglia? Ganglia is nothing but cell bodies of PNS. That is, cell bodies which grow outside of the neural tube. Will come to this term later, but now cell bodies of PNS is ganglia. If we go a little deep into this nervous system, basically it is classified into two groups, CNS and PNS. As we know, the term we should remember if we get the CNS is neural tube, and the term we should remember for PNS is neural crest. The CNS consists of brain and spinal cord, as we know, and this PNS consists of cranial nerves and spinal nerves. Cranial nerves are 12 pairs and spinal nerves are 31 pairs, as we know this. And the two types of neurons which is present in CNS are skeletal motor neuron and preganglionic axons. And the two neurons that is present in PNS are, here we came across the skeletal motor, and here it is sensory, and also postganglionic neurons. So postganglionic neurons, we discussed about this chromaffin cells in embryology germ layers. I'm coming across this point. This chromaffin cells migrate to adrenal medulla to form this postganglionic neurons of PNS. And next, myelin forming cells. What is myelin forming cell of CNS? That is nothing but oligodendrocytes. And for PNS, it is Schwann cells. So what are the difference between the oligodendrocytes and Schwann cells? Both produce myelin. The difference is oligodendrocytes, it can produce from any axon in this tract. But in Schwann cells, this produces in any axon in nerve. This is in nerve and this is in tract. So what do we call this distance between this myelin, either in tract or in nerve? This is node of Ranvier. So this part is node of Ranvier. The voltage gate channels of node of Ranvier are sodium. So what happens in the patients with the demyelinating disease? What happens is, due to the issues, this sodium distributes equally onto this, and the impulse passing procedure will be low. So next, if we come into the development of nervous system, how it develops? First, the process of neurulation takes place. When it takes place in third week. This both CNS and PNS, as I said, they are derived from neuroectoderm. And also we came across the term called as the notochord. I'll show you the picture. What it does is, it just induces the overlying ectoderm to form neural plate. That is neuroectoderm. So what happens by the end of third week, this neural fold grows in the midline and fuse to form neural tube. I'll show you picture. So here are the pictures. What I told is that neurulation occurs in third week. What is neurulation? Neurulation is nothing but the folding process of this neural plate into neural tube. That is neurulation, and we can see here clearly that notochord, I discussed about this term. This induces the formation of nervous system notochord. This inducer is called as the sonic hedgehog, and this, what it does is, this notochord overlies the ectoderm to form this neural plate, which is neuroectoderm. This overlies the ectoderm to form the neural plate, which is neuroectoderm. And next in third week, by the end of the third week, what happens is, these neural folds grow over this midline and fuse it to form this neural tube. You can see here in this picture the differences. And now this has alar plate and basal plate. Alar plate is sensory and basal plate is motor. These two are differentiated or separated by sulcus limitans. And notice this differences in this first neural groove, and neural groove grows over the midline to form this neural tube, and these neural crest cells join together when alar plate and basal plate is formed. And next, these neural crest cells are here, side of this neural tube. This is the picture at day 22. What happened here is this neuropores closes. This closes takes place in a particular days, that also in different ways. I'll tell you. First, what happens is this middle one closes, middle neuropore of this neural tube closes, and rostral end at day 25, this closes second, and caudal at day 27, this closes third. So actually first how it should be is rostral, middle and caudal, but neural tube violates the rule of growing from cranial to caudal position, because this middle neuropore closes first, and we can see some defects in this process, that is closure of that is irregular closure or not closure of this rostral neuropore causes anencephaly and polyhydramnions. We'll discuss about these terms in defects later, causing and increased alpha-fetoprotein and ACH. I'll tell you this at this end of the video. This caudal neuropore, these defects are spina bifida, which is very important. So I told you what is ACHE. This is nothing but acetylcholinesterase. This is the enzyme which catalyzes the breakdown of acetylcholine and other choline esterases in body. And what is alpha fetoproteins? AFP, that is alpha fetoproteins. These AFP and ACHE these levels are important for the detection of these diseases. Like ACHE defects comes when neural tube defects occurs. There is a difference in this ACHE, acetylcholinesterase, and AFP, alpha fetoproteins. These defects arises when there is some irregular or defects in development of this body wall. In here it is rostral neuropore. We can see this anencephaly and polyhydramnions with increase of alpha fetoprotein and ACHE, both alpha fetoprotein and ACHE, as it is body wall defect and also neural tube defect. And in caudal neuropore also we can see this alpha fetoprotein and ACHE, as both are body wall and neural tube. The examples of this alpha fetoproteins are gastroscasis and omphacele. You can Google it and see these images in newborns. And also this alpha fetoproteins are low in Down syndrome babies. So in next video we'll discuss about this central nervous system and also germ layers. We discussed about this germ layers and embryology, and we are doing this again. So if you like this video or if you have any suggestions, please like, subscribe and text me in Insta or mail or in comments, whatever you want. So stay tuned.